Hello, everyone. Thank you so much for your patience and waiting for us. We had a few delays on our end, but we're ready to get started now for a hotly anticipated event in this Lab Futures Forum. My name is Daniel Seiden. I'm one of the product directors here at Automata. And for those of you that aren't familiar with Automata, we're a lab automation business, and it is our mission to make automation a reality for every life sciences lab. And in our effort to progress on our mission, we have our platform product behind us here, which is the Link automation platform. It's a cloud-first, modular, scalable automation platform. And it is really our statement of intent in how we see the future of lab automation progressing as an open, scalable, modular, and cloud-first system. It was while we were developing Link platform that we started the concept of the Lab Futures Forum. And we really started this forum in order to progress the discourse on technology and its applications within life sciences labs. And it is really this event tonight that to me speaks to the quintessence of this forum in many ways, where we have our guest speaker, Tomasz Rzepka, from Artigen, who's going to be talking about Lab in the Loop, and also Rui Campos, my colleague and director of Link Cloud, who will be speaking about our platform product's role in that, in delivering on that future. So tonight we have an online-only event, thanks in large part to TFL's threats of strike action. So we moved the event to online only, and we really want to thank all of you for joining us tonight. We're going to be taking questions from all of you online after we finish all of the presentations. So do please feel free to register your questions through the chat, or after the presentations are over, you can raise your hand on the Zoom system, and you can then speak your question to the panel up here. So, without further ado, I'd like to hand over to my colleague, Rui.
Hi, everyone. Thank you all for joining us today. I'm going to take you more on the concept of Lab in the Loop. Here's a definition, typically, that you will find online about the particular explanation about what it is. But if we go into more like the exact representation of it, we're talking about the existence of the normal part of the process of executing something in order to get a science output. And much of this is derived by data being generated. So that captures the information that we really want to assess. And that data is then assessed typically by a human in the loop that is going to be looking into this data and identifying the best results that they want to take forward and which ones they want to apply some variation and really do it in a different way. And now when we're talking about Lab in the Loop is taking that from the human and starting to use artificial intelligence as the way that is actually going to derive those decision makings, or even learning from that and applying some additional decisions on top, and effectively creating this loop where all of this data that is generated flows through, identifies new paths that it wants to take forward, which things could be very interesting to pursue, and keep feeding back and forth, with no end in sight at this point. Why? Because from a data perspective, the volume of data is very important. Whenever we have an AI model that needs to provide decisions, those decisions have to come from a big piece of data that was used to train these models. And this is where high quality and high quantity data play a role. And this is where Automata really sits as one of the big providers that we can provide into our customers. Is exactly this, is our system is designed to provide you meaningful data in a very clean format, in a structure that you can really use and drive to feed into these models and to really drive AI forward. So if we think about it, automation becomes in Lab in the Loop is like a quintessent element that we really want to take forward for labs. So Here's a nice quote about people who do software should also do hardware, which we kind of do. But we're taking a little bit of a spin on it and saying be cloud native, because that allows you to then expand how you reach and how you coordinate things across the board, and how you bring AI into the loop with this, and how you bring also the facilities of running things, not just for one single work cell, but for multiple work cells where you can deploy simultaneously across the board and achieve that amount of data quality and data volume much faster because of that. So here's a little bit of a visualization about how we operate our system, where the physical system resides. And then there's a digital platform that enables all of the coordination of all of this through a very simple format that correlates and keeps the information about everything that happened in a format that is very well structured and very easy to digest by an AI solution. So things that we're seeing across the board in customers that we've been talking with is we're seeing this new lab standard of previously it was driven by people driving things. You know, we're now moving into this new format where automation comes first, and customers are coming to us and saying, look, I want to drive automation, and what are the roles that I need to put in place? Because it's no longer about the people that are going to be in the lab that they are expecting to operate these things, but it's more like, you tell us, how much can we automate and how much can we really take forward? And what are the roles that we now need to bring into this new approach that we want to bring across? The rise of AI being in as part of the equation, because now we're defining new roles. Why? Because many of these decisions could now be achieved by AI models that can really make something more meaningful in terms of speeding up time to market and generation of this IP. And turning these labs into factories, because if you think about the data volumes that we now need to have in order to feed into these models, you don't have a single work cell. You have multiple work cells, and they're working 24/7. So you have to account for these situations where everything is running nonstop, and it needs to continue to go and progress in that mode, and it needs to be running unattended. So that opens up other possibilities and other great things around that. And how do you connect all of this data and link into these other systems, which is where our cloud design and also our solution, even of what is running in-house, really ties into. And this is what customers also want, is like, how can I get that data faster, more structured, and how can I bring it back into my systems in order for me to process that? So these are all of these things that we're seeing, and the rise of many of these big topics. But also like the traditional role and how it's evolving in terms of automations. Like data scientists are now a big part of what you're seeing in the industry, as well as technicians are changing a little bit. So you're no longer managing things in the old ways where you're just thinking about 12 samples that you have to process in a day. You're now managing inventory, much like a supply chain system, where you have to account for all of these different samples, depending on how many samples you really want to process and how many reagents you want to have, and the discussions with different suppliers. And how do you ensure now that you have like a robust supply chain that you can drive these things across, as well as a research scientist? Because once you take the data scientist and the research scientist and you start to pair them together, that's when magic happens. So all of these things are starting to lead into a different approach, a different mindset, where a lab manager is more managing this kind of huge supply chain system. They're managing this data pipeline. They're managing the resource management of people that are going to operate the system and when things need to happen. So when we see all of these great things, this is where we see great partners. And these great partners are also the people that are helping us establish and lead the charge on this front. And in here, I'm going to pass on to you, Tomasz.
Thank you very much. Yes, and thank you very much for introduction and for like inviting me here. I'm very happy I can partner with you today to talk and discuss how efficient, automated laboratories are in fact friends to the machine learning and artificial intelligence world and as we see it now, because most of modern biotechs, or maybe tech bio specifically, believes that to improve the likelihood of their drug discovery, they really need to leverage to the full extent the power of the data they have. For that purpose, they build robust infrastructure around this, both wet lab and dry lab, in order to generate relevant readouts, but also to extract significant scientific insights. And this whole vision is very beautiful, and it's really powerful if you can apply it and build it from scratch. Nonetheless, the reality in many companies and in many places is a bit different. And it's not really something unexpected, because drug discovery is just insanely difficult. So you first of all focus on your science. You want to make it work. However, just after a few months in, you are basically left with your scientist wasting time to go through the data points, to manage the data, the data they have collected, to go through endless Excel files and be lost there. So those super intelligent people are not doing what they should, are not focusing how to interpret this and how to take conclusion out of it. On the other hand, there are already many working AI machine learning tools that can really improve some of the applications. Nonetheless, it's not really trivial to choose the appropriate method for your specific case. What makes it basically challenging to bring this beautiful vision of the modern or tech bio to all the companies. And what we have seen that the first step that in fact can effectively make a significant change in biotech or an other life science company is an attempt to implement an AI lab loop. And I think the basic definition is more or less known to most of us. So integrate AI to improve how the experiments are designed and carried out. In a feedback, you are getting better data from your scientists. Therefore, the AI models are getting more accurate. However, I think, and this is what we are seeing in our project, that what is really interesting lies behind and beyond this very polished definition. And it's really within the practical implementation, how those AI lab loops are made in practice. So what I wish to do today is to share with you a few examples how as Arigen we help a few companies to set up such AI loops. And I would like to a bit argue, discuss, and highlight three major aspects we feel that are the most critical to make it work in the long run and bring tangible outcomes to the component, to their drug discovery program. So let's start with a project where our partner had a very interesting drug platform. So on one hand, they have been working with multi-specific, highly potent drug. In addition to that, they were developing a platform to design those protein polymers that they attached to the drug in order to mask its activity in most contexts and enable it within the environment where really the disease and pathology occurs. So like a really nice example of focused and targeted therapy. And one important part of this platform was to optimize the specificity of this whole modality. in practice, it meant optimization of a fragment of this whole polypeptide, basically optimizing a sequence of around 15 amino acids. It's not a very long sequence, nonetheless, still the space to explore was quite significant. And what the team from our partner done was a very extraordinary job to develop a really efficient, scalable laboratory setting with many automated steps that made it possible to really implement a very efficient screening experiment. So they basically were able to generate data for libraries of thousands or tens of thousands molecules and were generating millions of data points. However, the sheer volume of this data basically overwhelmed them. They couldn't really navigate through what they have been seeing. And eventually, those were only, the next experiments were designed only as a slightly educated guesses based on a very selective analysis. So a lot of this effort was a bit lost because anyway, the peptides libraries that were subsequently nominated for the experiments was not really optimized in any way. In fact, even their computational biologists tried to help. Nonetheless, they had quite significant difficulties to communicate with each other. And this was basically a point where we were asked to help. And the first step was to set up a reliable data management system. And I will skip all the technical details around this. However, two aspects how we approach this, I think, were important. Firstly, we designed this to be really flexible to any changes and any requests we could find for any unexpected needs that could came up that we couldn't plan before. The second thing was to align with the major expectation of the scientists. So to make the data really readily available as soon as the experiment was done, so that they could inspect this almost instantly after the run has ended. So like it make like everything efficient and bring a bit of trust. Nonetheless, what was the really critical step was to build something that we call AI Workbench. So to design and build a platform to bring both wet lab and computational scientists together so that they could learn to speak the same language. So the tech behind the tech stack we use it's not really important, I think. However, what was important was to build this according to, firstly, the business goals of all the stakeholders they need, and also having in mind what the target group of users was. So we really made an effort to make sure that biologists could play and explore and visualize well their data comprehensively. On the other hand, to make bioinformatician enabled to show their results in a simple way and also propose the predictions in an approachable state. And finally, to that they both could explore those data together and came up with the joint understanding what is seen there. And this can seem not as something very impressive. There was no fancy things behind. Nonetheless, this cooperation and ability to bring those two cultures really enabled us to build a system that allowed them to firstly plan very data-driven and efficient subsequent experiments. So when they calculated some costs in longer run, we basically cut almost 10 times the time needed for the analysis of the data and the design of the next experiment. We also significantly cut the cost of the just experiments that needed to be done, because in the end, not so much libraries were needed to be tested, and the number of iterations was much lower. And as I have said, there was no fancy AI behind. So in fact, it was a very good and old linear regression that like worked really well there. However, what I think really bring the value was that, and this is probably the first and the foremost condition to build an effective AI lab loop, is that to build a bridge between the dry lab scientist and the wet lab scientist, so that biologist believes that in silico approaches can be useful. On the other hand, the data scientists know how to speak in an understandable way and how to communicate their analysis. So this is the first point. The second point I would like to make about AI lab loop is the data itself. And as an example, I will give an example of high content imaging assay, which cell painting is one of approaches developed by the lab of Anna Carpenter. And what distinguished this assay is that it's really target agnostic. It allows to characterize multiple cellular components of the cell, which brings a completely comprehensive view of the cellular morphology. But probably the most important is the fact that it's cheap and it's very easy to perform. So basically, probably any molecular lab out there can do an experiment with a good enough microscope that will create such colorful cellular representation as in the picture on the right side. And all of this makes this assay a perfect way to adapt for a massive screening campaigns. And probably this was also the reason why it was chosen as fundamental modality for many of the largest tech bios out there. It's quite generic and it's super easy to be automated. It gives very rich information content about the biology. And also when coupled with machine learning and AI methods, it really shows impressive result to improve the drug discovery process and have an impact on the overall programs, for example, for the small molecule discovery. Worth to mention initiative is also the JAMCP consortium, led by Broad Institute, partnered by many big pharmas, where also Ardigen is a technological partner, which basically enables a public database of hundreds of thousands compounds, which can be a starting point for an exploration in this area and application of this assay. What is the specific impact? So basically, cell painting or a similar corresponding high content screening allows for a confident compound screening or prediction of compound properties of very multiple or even 1,000 different properties or assay readouts based only on a single experimental assay as a cell painting. So we are basically cutting cost of 1,000 different assay and replace this with only the one. And with this is a result of our own research that where we have proven that using high content skinning can in fact replace very well and are superior to any kind of predictors based only on the chemical structures of the molecules. However, also there are more and more evidence published in the literature that proves it to be a really efficient way to improve either virtual screening or hit identification for the small molecules. Also, where we as ArtiGen see it very impactful is when it combines in the standard lab loop approach where we design, make, test, and analyze compounds. For example, in the application for the toxicology prediction, where we can easily and much early screen much more compounds with this quite cheap assay to filter those that could have inferior parameters in the process. So the story basically teaches us that we need to confidently and carefully choose what readout we wish to measure in the AA lab loop. So firstly, it needs to have rich information inside so that it has wide application. But on the other hand, it needs to be cheap and it needs to be approachable so that we can generate those massive data sets based on which we can train our models. And finally, let's get to the hot topic of the biologics, where the recent Nobel Prizes spark a lot of interest. If we were to read the headlines of newspapers or even scientific journals, we could believe that we can find, for example, a new antibody as in the model here in the picture. So basically, we can go for a vacation, and AI will, as a black box, will just design a drug that will be effective in the clinic. If we look into a bit more detail, what we see is still a very impressive progress in this area. So, for example, Chroma is a methodology that showed that we can generate de novo protein-protein complexes that to some extent resemble antibody configuration. An algorithm called RF diffusion showed that after a proper fine-tuning, we can, in fact, de novo design nanobodies that have efficacy toward a specific target. And finally, a method called Alpha Proteo is able, in a quite systematic way, generate binders for a target. Nonetheless, none of those would be a good therapeutic drug candidate. We are still not there. Therefore, in one of our ongoing projects in our area where we are asked to identify new hits for quite difficult target, so the black box approach is not really the case. What is happening is more or less as follows. So we have all those brilliant new methods and tools combined together, however, customized, fine-tuned, and put in the context of specific biology of our target. In addition, we need to add all the plethora of different methods, both machine learning, physics-based, expert assessment, in order to filter, improve, and prioritize everything that came out of the AI method, so that to filter and pinpoint only those that have realistic and reasonable structures. Finally, well-established high throughput. Usually it needs to be automated. Experimental setup is critical in order to iterate, to learn, to explore and exploit all those propositions of the AI, because none such AI workflows will work and have accuracy on day one. So we need to learn in the process. And in the end, we in fact came up with really efficient workflow where this combination of the good experimental data generation and very complex AI workflow can really level up our hit identification. In some cases, this can be an order of magnitude faster or more with higher probability. Nonetheless, still the final success rate, the realistic success rate which we can achieve, if it is 1%, we will be super happy and we would be very lucky to get there. So we still need high throughput screening methods, good laboratories in order to really leverage the AI to find the good hits in the experiments. So this constitutes the final point of our practical lab in the loop. AI methods that are used and applied, but with very deep domain expertise and knowledge of the experimental setting to make it efficient in like practical drug discovery program. And I think this basically are like final examples, and to some extent it showcase area that we as RDGen, as an AI CRO company, do for different biotechs and pharma companies, and support them in their mission to improve the probability of drug discovery. And as a company, we are doing this for almost 9 years currently, and we helped more than 100 clients. And in short, our main aim is to enable AI tools and capabilities to our clients, democratizing AI for the life sciences, at the same time building robust infrastructure that supports data-driven drug discovery.
That is great. Thank you so much. I'm thinking there was some questions online, so maybe we can start with those, because I believe everyone will be like raving to get some of those questions ahead of time. Shall we go through a few of those?
We kick off with the first one. So the first question we had was from Yaoyao Zhang. I'm sorry, Yaoyao, if I'm mispronouncing your name, but she asks, can you please provide a specific example of AI Workbench to help the scientists analyze data faster?
So the analysis itself, this is probably a joint conversation between the both of us to answer this question, because from our standpoint and the way that we do the automation is by accelerating the throughput that can be achieved and also providing the data in a very structured and repeatable format, and providing every single data point that can be collected. And even more, if there's sensors and other things, we can tie it directly there. And it makes that automation repeatable every time that you want to run and how much throughput you want to process through it. But I think that it's more of a mix. But how does that jump into the scientific side? And how do you make the best use of this data when we're talking about a workbench?
What I think is important here is to really understand the data modality you are working with, and then to properly choose the right tools to firstly visualize your data. And this can be, for the bench scientist, this can be super helpful if we find a way to, from a statistical point of view, it can be something simple like PCA or an other visualization method, but can allow them to look at their data as a whole. And for example, show which data points or which modalities they are testing are similar to each other, when which are far away, maybe what areas of the space they didn't explore, what patterns are within those modalities or those observations that they have already looked through, and which are efficient, which are not. So I think it's quite a journey and some kind of communication between a good data scientist and bench scientist to figure out what will be the most useful. And what we have seen in our project is that there is this eureka moment when they see the right visualization and can really, based on it and based on their really extensive experience and deep knowledge with good analytical tools, draw the scientific conclusion and say, let's then try this. This pattern seems so significant, so maybe let's amend this and check how it will work with something they know. So I think, I think this approach would work the best. Surely, there are some cases in which there are already established way to do that. But I think what is the most interesting is in the places that we are, as a life science industry, we are exploring that are very new, and we can really find something very, very novel and breakthrough.
Yeah, from our perspective, there's also a couple of things that tie directly and easily into this, which is you can define the workflow, you can define the thing that you want to execute, and you can define it with parameters so that you don't need to change that workflow going forward. You can take those hints, you can take those fine-tunements that you want to achieve for the next run, and you can apply it directly and just run it again without having to validate, revalidate, go through all of the process that you probably need fundamentally to run it, but you're just tweaking what you're trying to achieve, which touches directly on what your output could drive into this.
Yeah, so basically it's about you are able to control all the variables that are in the system. And if you are having automated lab, you can basically put it to probably 195% of the controllable, because you have most of the variables out of the equation.
Yes. I'm hoping that answers that question. We have a few more. Maybe go through.
Yeah, Spencer asked us, and I think that this question was directed at Automata, but maybe also to Tomasz. It was mentioned that it's important to have your software to run in the cloud for the purposes of being able to run models on it. Why is this so? Context, he recently interviewed for a data engineer role at a company that didn't use cloud architecture, which surprised him.
Well, to be honest, is actually quite interesting, because we can do also outside of the cloud, because we partner with NVIDIA and we can embed directly into the work cell. So it's not a constraint. But when you're trying to do it at the high volume that we were speaking about, that you really need to feed into the AI models, the likelihood is you're going to have multiple work cells. And if you're going to have multiple work cells, due to power requirements, in many cases, you will have multiple sites. And with multiple sites, connecting all of this together, either you have a very complex infrastructure that really can take the high volume of data that you do it, or in many cases, which is what we're seeing today, everyone is shifting into the cloud. The fact that our software runs both locally and in the cloud enables us to achieve a better correlation with these customers and a better direct integration to the ways of working that they have, and really feed everything directly there, which speeds up the entire development and delivery of the types of algorithms and the type of approach that you're also taking, Thomas.
Yes, I completely agree. I don't think that there is any specific, let me find the question once again. There is specific, yes, there is any specific preference towards, or like there are many advantages of running this specifically in the cloud. Currently, there are many solutions, both we run locally and in the cloud. It should probably mostly depend on your use case and what are your specific needs, and to adapt the solution to your case. We have clients that are working completely on their own local infrastructure, and it is efficient for them, for example, from cost perspective. On the other hand, we have others that needs to be very flexible, change things a lot, or have many remote teams. Then the cloud is the solution. I think both, there are instances and examples where either would be good. What is important is that to adapt this so that it feels within your priorities and what's your goals.
Yeah, from our perspective, we can run it directly as part of the loop, even during the execution of the automation workflow, because working with NVIDIA, we can embed these types of machines directly into the work cell. So there's no clear limitation there. What we're saying is if you're doing it at high volume, typically the cloud is the best option. And the way that we have integrated things, it enables us from the start.
It's sort of like saying, I want to put petrol in my car, so I need to build an oil refinery. It's much better to just lean on the larger network out there.
Yeah, ideally.
Yeah. David Brown, to Tomasz. We've discussed the outcome of the experiments as though it's perfect. No errors, no high or low controls, no automation is perfect though. What happens if a power delay causes a plate to run after sitting through some evaporation, skewing your results? Is your QC before your AI ML or after? And what do you learn from your controls?
I think, question. Yeah, perfect question. Surely, it was very simplified, and we are seeing a lot of different technical problems, but also biological variation in the data we observe. So surely, QC is a must-go, both at the level of very low-level data pre-processing, but also it's very important at the final step to validate whether what we are seeing from the prediction makes sense in the general biological sense. I think QCs are so inherent within all our pipelines that we are running that is almost native to the software. And also important thing is that for many of those QCs, we are running, in fact, AI models to detect them as well. That's so you great. If you are able to track what errors have there been in past, you can then align your AI methods to be able to spot them better. And sometimes you need to remove your samples. Sometimes the experiment will not be conclusive if it is too skewed.
This is one of the points that I think automation really is a big value add, because when we're thinking about those types of scenarios and errors, by having a very structured approach into providing that data and collecting that data straight from the source and in a repeatable fashion, makes it easy to identify when errors happened or when your data is probably skewed because of some reason. And you can see the variation and deviation and make really the best use of that. And in scenarios where you can even have environmental sensors close to your instruments that also flow through the same data pipeline, and get attached into everything that you're doing, which we can provide with our work cells. This is the type of data that allows you to say, this is an outlier because the data point does not follow what I've seen from all of the other data points. So I'm going to remove this one and really focus on the ones that are driving me the right value. And I can really accommodate for that. So this is one of the big value adds that I wanted to point out, that doing automation really drives a reduction in that error and a better utilization of the data, so that your company and your customers can really take the best output.
And maybe just to add one example from a project we have been collaborating. So in fact, there has been an issue with around 30% of the data. So there was some flow in the whole protocol. Nonetheless, everything was automated. So basically what we were able to do, despite some flow in significant portion of the experiment, to build methods that could adapt those quite large experiments to the major ones. So in fact, we didn't need to remove everything and throw this out. But in fact, due to the fact it was automated, so to some extent this flow was controllable and was predictable. So we could align this within the whole experiment, and thanks to that, have more powerful predictions.
That's great. I can see the chat is moving really well.
Yeah. This question from Mohammed. Do you want to read that out?
Yes, of course. Samash mentioned domain-specific knowledge, which is essential for each specific use cases. However, for cloud labs, where there might be interest in universal lab in the loop, what are the opportunities and technical challenges for those types of applications?
I don't really think that cloud labs change anything. So currently, if you wish to use any kind of more sophisticated AI method for a practical case study, you need to be able to customize it according to the domain with which you are working. And this requires, you need to be good structural bioinformatician in order to understand what AlphaFold is doing and how to tweak its parameters to be useful for your workflow. And then I would believe that for the cloud lab, you need to know what kind of experimental design you will submit there in order to get meaningful results. And there are so many parameters that you can set up for an experiment that you need to be an expert, and it doesn't really change anything.
And this is where you can help a lot, Tomasz, because of the knowledge of all of the projects that you already did, that you can help people really gain insight about what they can recode, calibrate, fine-tune in order to get the best outcome.
David Brown writes in with a follow-up question. He says, Tomasz, thank you, by the way. Can you bucket the clients that you've worked with? How many were ready to start at scale, multiple work cells, versus how many are startups beginning with a single work cell and planning to scale?
That's interesting. I think it's changing currently, and all the ongoing commoditization of machine learning tools, in fact, allow to more startups and rather small companies that are really focused on their scientific idea to come up and have this ambition to build something that will be scalable. Even if they have quite limited budget, they are quite confident that firstly, then they can automate their very proprietary scientific assay, and then use AI to scale this up so that to have much higher probability to get it validated and move forward. And yes, we are seeing more and more of those examples. Nonetheless, I think majority of the companies we have worked with in like similar cases, those are mid to large biotechs or mid to mature biotechs that have already some history of the in the industry, have already some system set up, have quite significant lab there, and yeah, are getting overwhelmed by data.
Yeah, I can say from an automation perspective, we are working with larger organizations, but also startups. And on both sides, we're seeing an intention of going to multiple work cells. And in some cases, we're even deploying like 2-digit work cells in some cases, which is quite interesting.
To put a little more color on that, generally we're not their first work cell either.
Yes.
And
that's
often,
true.
and at the least, we are working with individuals that have significant experience with automation within liquid handlers. But I think looking at these types of applications, these are very experienced individuals and organizations when it comes to total, and workflow automation. That's a fair characterization, would you say?
I think I'm unable to answer right now. So I don't want to lie, out of the hand. I don't have. I don't think I have an opinion right now.
Fair enough. Fair enough. We have Scott, who's asked a question for Automata. Can you discuss a little bit how the tools, Link WorkCell and/or AI workbenches, have been utilized to operationalize lab automation in commercial manufacturing lab environments to execute continuous process improvement?
Well, we're more focused on the automation itself, not the end game that the customer is deploying into. Although we do have a big customer success team that is just focused on outcome and ensuring that whatever the customer wants to achieve is doing there. But maybe you have a few views that you can share on this as well.
I think to me that in that question, the word that stands out is manufacturing.
Yes.
We don't get involved in manufacturing spaces per se, that are producing commercial consumables. We really are working on manufacturing at a different level. That's sort of more on the exploratory side of things rather than the finished product that goes on a shelf out the door of the and into a truck. So we definitely, that word manufacturing can be a little bit loaded, but if you look at it from the sort of R&D manufacturing of data, absolutely.
Yes.
We're about scaling that up to in a very manufacturing-like manner.
More the research itself,
Yeah.
and more like the small scale that can lead into a vaccine that you can apply and test on trial runs.
Yeah.
But not as much on the drug manufacturing as a whole, which is a different type of scale and a different type of problem also to address. And in those cases, although we haven't directly applied or deployed one of these for those types of scenarios, we have worked and with customers on assessing and identifying how we could approach it. But in those cases, the AI is more for operational reasons and optimization.
Mm-hmm.
So the problems are very different than probably what we've been discussing so far, which is more research related and more like driving models of AI models or decision makers in order to feeding and getting more data outside of that. So I'm hoping that this answers Scott's question. I'm not sure.
I think it does. And I think that particular point you just touched on, where it's more about operational purposes, is very much what you're seeing in heavy industry.
Yes.
Which arguably in many ways is more advanced when it comes to automation and applications of AI than even we are, because we're dealing with a very different problem space. They're dealing with something that is a very well understood problem space and an optimization of that problem space. So you do see that very much applied there, but it's still not. I haven't really heard that narrative in our space.
So like in terms of manufacturing, we're in fact seeing some kind of interest in trying to rewire this process a bit and find like new ways forward. How I see it now, I think they are in quite good spot, like to optimize how they do process right now, but possibly there are some other routes that are a bit out of their out of the scope how they are looking at this process that could really even more level up. So how they are doing this now, I think it's almost to the extent. However, even with vaccines manufacturing, I think there are even some research and or AI methods that can really even boost this up. And we, in fact, had such engagements this year, and they seem very promising. So I think also in terms of the automation, it just needs some kind of paradigm shift. How those people there look at it.
Yes, I agree. And we're seeing a lot of that happening, especially since COVID. I think automation became a much deeper knowledge kind of field that everyone started to look into because of a necessity. And we're playing into that game of driving it forward. I think we still have like a couple of questions, and then we could probably use some of the other ones that we had also in place.
Yeah. I'm going to call this person's name Ola because I don't want to embarrass myself in my pronunciation of it. So I hope you can I can apologize to you, Ola. So Ola asks the very poignant question is, have you got customers recently?
Well, we do. We actually are in a great position, at least from our side, where we decline customers because we're trying to reach the right ones at the right time for our scale purposes and the journey that we're on. So we do have very interesting use cases that we're working on. And I'm sure you do as well.
Yeah, I think we are currently fully packed in terms of the engagement of our company, and we are like almost 180 people. Also, I think just this morning I got info about new deal that will be signed. So yes, we are getting new customers.
Excellent. It's great to hear. We have another question here from Thomas. Thomas says, I'm a wet lab scientist in a small startup looking to automate some of our assays. We have a LIMS, a confocal, and liquid handler. What kind of coding or IT skills should I learn?
This could be approached from multiple angles. Maybe I can start from the automation perspective. Well, it's up to you. Our system has both a UI so that you can rig it, design, and deploy and run it and get the data directly integrated into your LIMS system, or even other system if you want to then export and create a data lake of your own. But we also have an API that you can integrate and generate your entire workflows and even design them on demand dynamically through our API and SDK. So we give both options. We don't limit anyone, and it's not just a start-stop of a run. We provide the ability to recode the entire workflow directly from code. So no limitations from this side, as much as you want to take it.
Yes, and from our side, so for very small startups, we specifically have this offer of some kind of computational onboarding or like cloud onboarding, where like for quite low fee, we teach we can teach you how to implement some of the standard computational environments within your workflows, and then enable it to you so that you can have like ready access to like the most state of the art solutions. So we sometimes do that, and we try to be like approachable, but on the other hand, does not have like a very steep learning curve.
Yeah, from our side, whenever we deploy, we always deploy it, and we have a strong IT team that provides all of the support required in order for that work cell to be fully functional. If you want to deploy it via our API SDK, the preferred option is Python.
Great. Should we move on to some of our questions for Thomas?
Yes.
I think we have a few.
Let's go through that.
Yeah.
So When we spoke earlier, you mentioned that we're not yet at the point of having a lap in the loop ecosystem, and that we're kind of doing these rather bespoke systems, which kind of touches a little bit into probably a little bit of infancy in terms of the growth of the area itself. So can you expand a little bit more on the distinction of a system that is more bespoke versus something that is more fully formed?
So I think the fully formed system would be something that the whole company or the whole organization have in mind this data-centric vision. They understand that everything focuses and circulate around data, like all the processes in the organization are set up in a way to support this process. All scientists, both wet lab and dry lab, knows that they want to contribute to this data-driven, data-centric vision and data-driven drug discovery. So I think this is rather question how a specific organization is set up and how people around think and in what direction they all go. So if chemists have in mind that our overlying goal is to have a very good AA lab loop, he wants to contribute to how the AA models work, and it's not only concentrated to make his molecules as perfect as possible. So I think this is a different mindset, and if this mindset is shared throughout the organization, then you will have this full-fledged system. On the other hand, for specific use cases, you can build those AA lab loops, but then this is rather a process that have very specific goal. You set up the limitation and you basically build firstly the tools, so for example, the software, but also some cultural processes and or communication like to smooth it up. Yeah, I would make this distinction, and I think in practice this is how it works in industry. So some companies really try to build this within the organization that are quite old one, and maybe a few of them are succeeding, but this is a long process. However, if you're starting with this idea, then you are basically building everything around this, and you are arriving at this state and don't have all those artifacts that are like behind you.
Yes, we're seeing like these kinds of trends of more established companies do have a change management process that they have to go through in order for it to really become ingrained and established in the organization. While startups are starting with that mindset from the get-go, and that speed that they're gaining of driving that automation, that data-centric approach, that drive in order to create this flow internally of usability and driving things across, really is the thing that is making them successful. Is that pace and time to market that is really driving them forward. And what we're seeing is, on the flip side, the other organizations are seeing how they're moving and the pace that they're driving this, and they're now trying to ingrain and instill themselves to being at a similar approach, which is quite fun to watch.
We have a question from Edwin, which is relevant to this concept of the ecosystem of Lab in the Loop. And he says, I work in GXP regulated environments. Do you provide computer system validation packages that cover data integrity? Have you validated an AI that is used in a GXP lab before?
That's quite interesting.
It is a great question.
I think this is quite a long journey to validate an AI for that purposes. We have built AI systems that afterward have been validated for the regulatory purposes, but we didn't do it like ourselves. most of our stuff is, and also some of the data management systems, we are also going into this direction to have a more comprehensive regulatory certification for many of the pipelines, even for like genomics processing.
I'm going to put you on the spot now, Thomas. How far in the future until we see this as a standard offering?
My personal opinion, if it will become more and more present, then we should probably implement even stricter regulatory and let's say real world validation, how they behave. So I think there is a lot to gain with AI in drug discovery in clinic and diagnostics. But on the other hand, we need to, all of those methods need to gain trust in the society, need to gain trust in the stakeholders. So I think all the marketed AI drugs that are out there, so they should go through even stricter regulatory and or like clinical validation to prove the point. And I think like with any like AI tool that will be deployed anywhere, this should be the case.
That's great. Great. Shall we move on to another question? What are the biggest misconceptions biologists or chemists have when working with AI-driven tools and closed-loop systems?
Yeah, sorry.
Yeah, sorry. So I'll repeat the question. No problems. It's like what are the biggest misconceptions that biologists, not in there, I'm just running through one of the ones. What are the biggest misconceptions biologists or chemists have when working with AI-driven tools and closed-loop systems? Where do you see there's a disconnect?
That's a very interesting question. I think I don't think there are a lot misconceptions. There are rather a lot of reservation and a lot of disbelief. And what a lot of chemists, for example, says after collaboration with us is that they really value when we are showing them how those methods work in the backend. So like not in all cases or in all examples, an AI tool will solve the problem, but showing them what are the patterns, some kind of AI tool propose a new molecule, for example, or on what part of the molecule it looks to make the prediction. It helps them to understand how it works. And I think they don't really have misconceptions. They just either don't know how it works, therefore they don't believe it. And in the practical application, probably the main point that prevent them from using AI is the idea that they need to have a data set of big pharma size in order to start anything with AI. So it's more about maybe not misconception, but being brave to try and have some kind of curiosity to explore, even with smaller data set. And I think with all the firstly large public data sets that are being enabled and available, and all the transformer models that came out recently, it is more and more available to have a big public model, open source one that is trained on this massive data set. But this enables to use quite small internal data set for your use cases. So really bring it in-house. And I think, yes, so possibly this would be this major misconception, I think, that prevents some of the companies or some of the teams to adapt this to a larger extent.
So we're talking about education as a major thing to get everyone in alignment to understand like the possibilities of what we have right now. Second, the data sets that are available that could speed things up like quite substantially. But I would also say is we're still in the infancy of many of these things. So there's still very much like a low-hanging fruit that doesn't require as much of a data set in terms of the volume of a data set to really get to the outcome. And this is where I'm seeing also some startups focusing and zoning in is like, where's that low-hanging fruit that I don't need as much as a bigger data set because I can get to it really fast. And I just need to have enough automation and this 24/7 cycle that is going to get me across, build me that data set and then prove my point.
Yeah.
Yes, and in fact, with AI, you can scale it up from very low throughput to like maybe mid throughput, and this like probably increases your chances of success with your idea of such small companies quite significantly.
It's quite
So
interesting.
it sounds to a certain extent like what you're describing is, yes, education, but also the tools for visualization and understanding, these tools for communication.
I would even say interpretability. So like being able to show what's inside of all those algorithms. So in the end, those are just computational tools, right? So and in most cases, for the application to chemistry, biology, bioinformatics, we are already quite well equipped to try to explain what's happening inside. And those approaches are like quite gives quite good result, and it really also bring trust because, for example, if we are building a predictive model for some kind of molecular biomarkers to predict a disease, and we are saying it has like 90% accuracy. This can be taken as granted, but this is just a number. But then if we came and say, hey, because this model in fact combines those three biomarkers together, and if their level are somehow aligned, then this prediction is made. So we are giving a biological story behind the prediction. So then it really clicks, and also it really helps to adapt this and believe it.
Yeah, that makes sense. Then the only barrier left is actually getting to you. Yeah.
So having an interesting question about the bottlenecks, because we're dealing with life sciences. It doesn't really behave as you would expect if it's just a digital system. And where do you see the current bottlenecks in order to getting to the outcome? Is it the work cells and the capacity that you can process things? Is it the processing of the data that comes out? Is it the design of the experiment? Or is it anything else? Where are you seeing like in the projects that you're running that the biggest bottlenecks are at the moment?
I think that the process of generally drug discovery, like hit to lead or lead optimization, this is quite well understood and like well optimized. Whereas the bottleneck is mainly at the de novo generation. So getting something for those really difficult targets, or for some of them that we have no idea. And here probably we need completely new scientific breakthrough to crack this. And the second thing is probably rather a biological bottleneck is the transferability of our disease model to the clinic. And I think this is really the major problem where most of the resources should be put, because this is what really drives the low success rate of the drugs in the clinics. And then here, I think like automation is probably can help a lot, because with all those new disease models like organoids, IPCs, you need to generate them on a scale to be able to explore which of those ideas could be relevant and useful to bring us more relevant disease system where we can then test compounds and build around it. If we crack this, I think then everything else is mostly engineering problem, and which we can pretty well solve with like a systematic process.
Quite interesting.
We're coming up on the 8:30 mark, and I know you have other events to get to tonight, Tomasz. Thank you so much for your time. Maybe we could take a couple more audience questions.
Yes, I think so.
And then wrap up. Great. Well, we have another question from David. David says, for Automata and Tomasz, a lot of this automation is based around liquid pipetting. How well does this play with microfluidics, cell sorting, and other selection approaches like cell sorting or phage display?
Tomasz, do you want to kickstart on this one? Because it feels to me that it touches more on the type of projects that you've been working.
I think I have a little biological expertise to be able to answer with this with like, yeah. Enough, you know, yes. So sorry, I will pass on this.
So from our perspective, we deal mostly with the automation across multiple instruments and ensuring that the easiness of creating the entire workflow end to end is the main thing that we're trying to speed up and help our customers with. In the same vein, we provide all of this tooling, but also the ability to work with us on ensuring that anything that you need to achieve from a liquid handling perspective, that we either partner with the right partners or provide some support to get that across and to run it through our system and generate the data and then export it into the right systems and connect all of these dots that previously were not connected. So our main point is not really the science. We leave that to our customers for them to define it themselves. Our focus is on automation, but this is where we partner with people like Tomasz to really help customers take it to the next level by using AI and really creating projects that drive meaningful value. I know that it's not the answer that David probably wanted to hear, but I think that's the context that we can bring for this one.
Absolutely. And as the topic, or at least a great deal of the topic of tonight's discussion is about the volume of data needed in order to make these guided decisions. That's why scalability is very much at the center of the design of our system and how we feel the future of automation systems within the lab needs what it needs to speak to, because you can build in the redundancy of devices in order to really scale up the throughput and generate that higher volume of data within a confined space. So we're very much betting on that as being a key characteristic for serving this need.
Maybe take the final question that we have there.
Sure. We have a final question from Leo. Leo says, how much of a focus is interoperability in your pipeline? What types of interp methods are you actually using?
So I think it's for me. I think in 90% of projects we have done for last year, we included the interpretability modules. So we try it to be a standard, right? And the interpreter, like the method we actually use, this basically depends on like what type of the input data or what type of modality. We have this this can be one of the standard ones like Shapley values or integrating gradients. For standard neural networks, nonetheless, we have also been developing more focused and more custom interpretability for more like graph neural networks for chemical applications. But then we really try to adjust the purpose of it with what the user wish to see and or wish to understand about how the AI works.
So I know that Scott was the last one, but it's just a 1 minute answer for Scott. We have a library of different instruments, and it's already a large library that we have today. And we're even next year going to be launching the ability for anyone to build their own drivers in our platform. So if you want to reach out and know a bit more, we're happy to take you in that conversation and really help you understand where our direction is and where we are right now. So I think that wraps us up.
It does wrap us up. Tomasz, thank you so much for your time tonight.
Likewise,
It's been
thank you.
absolutely fascinating.
Yeah, thank you a lot for
Amazing.
invitation. Like,
Thank you.
It was great
And
discussion.
I'd also like to thank everyone who joined us online.
Yes, thank
we
you
really
so
appreciate
much.
your questions and your interaction with tonight's event. Tonight is the penultimate event of the Lab Futures Forum for the 2024 year. Our final event comes in December, where we're going to be doing a bit of a Christmas social in partnership with the London Lab Automators Forum. So do pay attention to the Automata events page and sign up for that event. It's going to be good fun. And thank you all very much. Good night.
Thank you all.